session of the conference, and I hope everybody's sticking with us. This is, again, Nathan Peck, Cure VCP Disease. We've got an awesome panel here. I will admit this is we didn't have much of a plan for this session because we wanted to leave it open based on some of the themes from the conference. So we're going to have more of a fireside chat. We welcome all the listeners, everybody in the session to join in, ask questions, post your questions. We're just, we've heard some kind of main themes out of this session, through the whole conference, and this might be the time to kind of extract it a little bit more, or discuss it. So we tried to balance out the session with, clinical and preclinical, because we've had a lot of different topics in that regard. But let me introduce our panel first. We have Dr. Tahseen Mozaffar. He's professor of Department of Neurology at the UCI School of Medicine. Then we have Dr. Chris Weihl, professor of neurology at Wash U School of Medicine in St. Louis. We have Dr. Jordi Diaz Mendez, professor of neuromuscular disorders at Newcastle. And then we have Dr. Michele Arkin, department chair and professor, Department of Pharmaceutical Chemistry at UCSF. And so again, this is just kind of wide open. I think what I wanted to start with was, from the conference, were there some immediate needs or things that you see that need to be addressed, or maybe even just a takeaway from the conference in terms of VCP, your understanding, or what might need to be addressed? So I'll leave it open there. And I know some of you have jumped in and jumped out, but I mean, based on some of the conversations, what do you think? Chris, come on, you're chomping at the bit.
All right, I'll start. I think I'll start with patient heterogeneity and really understanding why there's so much patient heterogeneity. And the reason I bring up patient heterogeneity is because we just listened to a great talk about kind of the natural history, or the really just a cross section at this point, and just hearing about the heterogeneity in those patients. And I think one thing we didn't talk about, at least I didn't hear it discussed, was kind of, if you were to design a clinical trial, would you use a subset of those patients? And so that's where I think the heterogeneity becomes important. Would you want, I know, we all want to treat every patient, but we want to make sure a therapy gets adequately evaluated. And so I guess understanding the heterogeneity, understanding what leads to that heterogeneity, whether it's a genetic change, a biomarker, whether it's presenting with an MRI finding first, how that then can be used to then stratify patients for a therapeutic trial. And I think, and so I say that mainly because I think that spans multiple interests in some ways. It spans what's interesting about genetics, what's interesting about modifiers, what's interesting about MRI, what's interesting about protein activity, and how do we then move that into therapeutic development. So that's kind of my
and Chris, I think you're right on on the point on this one. I mean, part of my challenge excuse me, is that, in a disease where you can have a stable patient with myopathy who may all of a sudden change the disease course because they develop motor neuron disease or develop frontotemporal dementia, is going to be challenging. We really don't understand the natural history of this disease and how some of these factors, let alone the genetic and the modifying factors, affect the disease. On that, and it's for a rare disease where the patients are spread throughout the country. How do you carry out a natural history study to get meaningful trajectories on how the disease course happens is a very important question that we need to address. And I know that I've spoken to you about it. I've started this discussion with Jordi, who's been doing it, at least in Newcastle, and then using biomarker as to inform the natural history study, be it the MRI stuff that imaging stuff that Jordi is working on, looking at some biochemical biomarkers, whether, I mean, we have an interest in TDP-43 or some of the lipidomics to look at natural history study and how those biomarkers behave with time. But I unfortunately, at least my personal opinion is that we may not be ready for a meaningful phase 2 or phase 3 studies till we fully understand what the natural history of the study is and how do you predict how the patient is going to do. And I think, Jordi, you're really eager to add to that comment. So.
No, yes, of course. Thank you very much, Tahseen. So this links with the reality is that it's not only having biomarkers of disease progression, it's also having biomarkers that can predict how patients are going to behave in the next, for example, 5 years. That's something that I think that we don't have. So the reality is that when you face a patient, of course, you can comment to the patient that these are the possibilities in terms of symptoms. We know that based in in what has been published, we know that this amount of patients will develop myopathy, this will develop Weill disease, etc. But it would be great to identify some disease predictors. So we can talk about biomarkers, serum biomarkers. You have mentioned MRI. Maybe there are others that we don't know or that we are not used to look at that can be helpful. And I think that it's different to identify biomarkers that follow disease progression, which is one topic, and another one is something that can predict what will happen in the future, which is a different one. And I think that for what you were saying, Chris, in order to stratify patients for clinical trials, what we exactly need predictors, some things that can predict disease progression. And of course, for me, well, this is an opportunity to talk about MRI. Of course, I am a passionate of MRI. Yesterday, we just presented some data on the utility of MRI for the diagnosis. But as you know, quantitative muscle MRI have many different sequences that can be helpful. And not only that, I think that in a disease where there is protein aggregation in the muscle, there is also room for testing new PET MRI scans and new opportunities in order to identify the accumulation of these protein aggregates in the muscle. That could be also very nice. So having said that, I'm going to change a bit the topic, and I think that after listening all the talks, I just still think that we need to clarify what is the function of this protein. I'm sorry to say that, but I think that there are many different theories about what the proteins do. So we have a clear idea what for sure it does, but I think that we have heard that it's involved in lipid metabolism, it's involved in protein that metabolism as well, etc., in the ubiquitination, etc. But I think that we still need some clarity, and I would like about the function of this protein. I would like to know the opinion of Michele regarding that. Do you think that we still need to clarify what is the role of this protein in the different tissues? Is exactly the same role everywhere? It's different in the brain, in the muscle. It's different in the muscle, in the bone. What do you think about that?
Well, definitely everybody here will have something to say, and I hope the audience jumps in too. I think that it does a lot. It unfolds proteins. So in a way, it does one thing, but it does it on lots and lots of things. So those things are going to be different in different cellular contexts. Age, other genes, muscle, different cell types. So I think that's maybe where that complexity comes into. It's that intersection of having lots of substrates and also lots of adapters that are helping it select the substrates. And yeah, so I think we do need to understand what it does, but I'm not sure how much biochemistry, I'm a biochemist, but so I'm not sure how much biochemistry is going to help us versus pigeonhole us. We always have to be thinking to cell type, and then also we can't think like, well, I do this, so I have to think about this, because we know somebody else is seeing something different in the same cell. So we have to go up and down in the layers of complexity, I think, to answer your question. Chris, you probably have something to add. or disagree.
No, I was looking at some of the questions. I'm sorry.
Oh, sorry, I didn't mean to put you on the spot.
That's okay. That's actually Eddie was going to ask a question. I'm sorry. He had
Yeah,
an
please go
important
ahead.
question.
Yeah, Nathan asked me to jump on. I something that wasn't discussed that I'm very curious about are your thoughts about VCP related IBM versus sporadic. Like, how much can we take from understanding VCP mutations apply to sporadic disease and vice? So that's on a basic level, but then on a more clinical level, how similar are these diseases? Because I don't see patients, I see their tissues. And is that a possible way to increase the patient population for potential VCP therapies?
Yeah.
Presumably, I mean, like the disease specific, the mutation specific therapies obviously are not going to be used in sporadic IBM, but if VCP inhibitors or activators or something might be applicable to sporadic disease and get just bigger numbers.
Yeah, I mean, you're speaking to what I do. So, I mean, I think, one thing to know is that they are very different diseases, but, there are patients with sporadic IBM that have been identified to have VCP mutations, whether they really have multisystem proteinopathy and I just misdiagnosed them as sporadic IBM, I'm happy to have that discussion. The point is that they clinically look indistinguishable in some cases. As far as their pathophysiology, I think it's unclear. They have very, very distinct similarities at the pathologic level, as you know, as a pathologist, that can mean something and it cannot mean something. I mean, and so I think one distinct difference is that inclusion body myositis has inflammation associated with it, whereas inclusion body myopathy, which is what we see in VCP disease, does not. The truth is, and you may not realize this, there was a trial for inclusion body myositis using a drug called arimoclomol. Arimoclomol is a drug that increases the heat shock response. That trial was done in inclusion body myositis. The preclinical study for that was actually done in a model of VCP disease. And so people are actually thinking about what you're saying. Unfortunately, that trial failed to meet its primary outcomes, but that doesn't mean it wasn't a good idea. It just failed. So, but I think it's we talked a lot with that company about expanding their trial in sporadic IBM and said, hey, what if we got 20 patients with VCP disease and threw them in? That sounds like a great idea. The company wasn't up for it, but I mean, I think that's exactly right. And I would say the same thing to you about other forms of FTD. So we were very interested to find that a signature of VCP deficiency seemed to match what you see in progranulin patients, suggesting that is there some overlap there?
Yeah,
So-
I was gonna.
Go ahead.
Oh, sorry, Suzanne. That was my top question too, was how much that we're learning about these mutant-based diseases are going to be generalizable to the sporadic versions of those diseases. And it really gets down to what is our drug target. If our drug target is normalizing VCP activity, we have to demonstrate VCP activity is messed up in those diseases. But if we say, well, VCP is not the target, just like SOD is not the target of ALS, it's the inducer of the ALS, maybe we need a target that's not VCP, and that will be more transferable. Stephen?
So we can summarize that one of the things that we need to understand better is the pathophysiology of the disease then. Because we have been talking about biomarkers, biopredictors, we can put that name, and MRI, but now we are trying to understand what is the function on each tissue and the pathophysiology. You agree that this is still not known?
Right. So I think the fundamental question we still have not addressed, right? We still have so many functions, so many different cofactors. There are certain cofactors we have nothing no information of what they do. And for this disease, different 3 cell type, the most of study is muscle, and there are a lot of motor neuron, but there's very few done in bone even. So if we say, if resource is unlimited, put that out of the table. As a group, what do you think are the systematic way to tackle the question in unbiased? No, we don't pick up our favorite essay. We just say what would be able to advance the field and help the clinician, help the cell biologists, help the biochemists, structural biologists to work together to what would be the best dream team to make this make the progress faster. Yeah, Chris.
I mean, I think so. I mean, I think the makings of that are around, like here. Like, I think you're right, and I think the questions are, if we were to start with the issue of what is the mechanism of action of the protein as far as at a molecular level, which I think, you're well suited to figure out. I think the next question is, what is the effect of the mutations in different tissues? And then I think that will then help us identify some biomarker that can be used for, one thing we didn't discuss is kind of therapeutic biomarkers. So we need a biomarker that correlates with disease, but we also need one that we think will change whenever we have some intervention. And so it may not, as Michele said, it may not be VCP activity. It may be TDP-43 levels. It may be, global ubiquitination. So I think that then requires detailed phenotyping of patients, and then which is being done kind of through the natural history study. I mean, I think it's, I think part of the issue is, I don't know if we have a good therapy. I think people, we think maybe the inhibitors, but I don't know. And so I just don't, I feel like at some point we need a better idea of a therapy.
Right, but I mean, even I just feel that even back out from the therapy point of view, if we don't understand what is the fundamental issues, then the therapy we try, right? Because you have something available and you try to see if it works. But if we want to really understand the disease, would they, now we know, a lot of people are trying the IPS model, but we know even try to use CRISPR to make isogenic 9 for the same mutation from different patients, the result is different. So how many N do I need to do to get a meaningful result that can change that to help? Do we focus on the human patient bank? And if we do, what type of tissue will be more important to, and then it become a really big project to have enough resource to profile all of the sample and get, even to select the right patient cohort just to do the biomarker study. What's your opinion, everybody's opinion about how to approach that?
Yeah, I think when we looked at this, coming from a company perspective as looking at, potentially developing inhibitors, one of the big things that we notice when we look at the literature is there is not a fundamental foundation of the basic biology of the disease. There's not a paper that says this pathology is driving this part of the disease. And so I think there is some ambiguity as to whether an inhibitor would help. If you can't say definitively that the gain of function mutations of VCP is what's driving the disease, then you can't say that an inhibitor is the right, treatment there. If the mutations are causing disruptions in protein-protein interactions, and some of the talks at this session showed that when you knock down VCP, you improve disease. If you're knocking down VCP and you're improving some of the disease readouts, an inhibitor is obviously not going to be helpful in that situation. And so being able to have really well thought out experiments that can link a function to a disease pathology and whether or not that's actually what's driving disease in people, I think is a fundamental question that needs to be answered before you can begin to think about what type of therapeutic intervention would be useful.
So who want to answer me about what do we do if we want to find a biomarker? What is the, best approach to start?
To find what? A biomarker?
Yeah, from the patient sample. I know, IPS is one approach, but I just, feel it may not be the best approach.
I think serum. I think patient material is the way to go. I think the issue is, you could do some. I mean, you just said, you need to have 2 or 3 cell lines. You're going to need to have like 100 patients. I mean, it's even harder to find something that, it's not like there's one single thing that I think, it's possible you could find one single thing, but I don't have a good answer.
But I think what you said is really important. So you found it. Did I get it right? You said that you take the same mutation, make iPSCs from people with the same mutation, and you get different results.
Right. So if you do, we haven't do a lot yet because this iPSC is really expensive, right? So you do a few, and then you try to say, okay, we replicate, and there are different patients, but we did trace out the issue. Like some of the patients that we buy from this bank, they claim they are normal, or they claim they are something, but if you sequence deep enough, you find some mutation that is in the same pathway. So there are these background genetics that, as if the best approach might be, start with wild type and mutate to different known mutation, but then you rely on just one wild type. So there's this caveat of what is the, and then you say, let's go to clinical sample. Then it comes with how many N I need to get a powerful or enough information to make the study meaningful, right? We don't want to get other data and then we still don't know.
From the Alzheimer's field, you need not hundreds, you need thousands, if not tens of thousands of patients. It's very, very difficult, and even a plasma test for Alzheimer's is barely on the horizon. Maybe the iPSC. I'm intrigued that NIH has this new initiative where they're making. They all start with one parental line and they're introducing mutations and then reverting them back. So you could get the original control and the revertant cell line with the mutation. And I check with them. They're making, they've actually made already 3 or 4 VCP knock-in lines.
Is this the NIA one?
Yeah.
Okay.
Yeah.
Okay. Mm-hmm.
And that might help. I like the idea of the control mutant and then a revertant on one background, but it's the caveat. It's one parental line for that whole project. So if that one parental line has some weird interaction with VCP, it's going to be, difficult.
I mean, I feel like if we look at the muscle pathology, I'm surprised that I heard Jesse say that he thinks the pathophysiology is confusing, because I think it's due to impairment in protein degradation via autophagic pathways. I mean, I think it absolutely, that's what the disease is from. That's what's causing it. I think if you want me to find problems with endocytosis in that muscle, I've done it with hemo. If you want me to find problems with mTOR signaling in that muscle, done it. But I think the underlying pathophysiology that's causing the disease is related to autophagic dysfunction. The reason I think that is because of other inclusion body myopathies that have similar pathophysiology. And so to me, the fun part about VCP is that it's involved in all these different things, and I'm ready. I'm going to look at lipids. That's awesome, but I don't think that has anything to do with the underlying disease, and I don't think that is as much of a mystery as we're stating here personally. And I'm being a bit dogmatic on purpose, but
no, I think that's interesting because then when you think about aggregation or stress granule formation, any of these other, they have or lysophagy, mitophagy, these all have that kind of component. Would you scope, when you say autophagic dysfunction, would you scope that pretty broadly to include those things? Or are you really thinking of the autophagosome, lysosomal fusion piece?
I think it's the autophagosome lysosome fusion. I mean, that's the biggest thing we see. TDP43, I'd be curious to see what Eddie says. I mean, it's a striking, so I do muscle pathology. There's things that are striking features where you see it and it's unique. And when it's there, it's very helpful. But there's very few TDP43 inclusions. In fact, it's less, it's 1% of fibers. And so it gets a lot of, press, and everyone's like, oh, it's got to be well, no. The real problem is that there's degeneration of muscle fibers that are accumulating P62 and LC3. And so that's how I think about it. But is it fun to think about stress granules, and do I want to study it? Yeah. And
I guess what I was trying to articulate, maybe I didn't say it as well as I think, is what is driving that, I guess? Is it an overactive enzymatic function that's driving that an inhibitor could help with? Or is it protein-protein associations? And are those being disrupted, or are they being enhanced? And where I'm not as well read in this literature, so I don't know the answer there, and maybe the answer is known. But when I look at it from the outside, I don't see a definitive answer there. And I think one of the things when we look at it from a drug development point of view is we just look at the landscape of drug development, where they've had, these long-standing theories in Alzheimer's and Parkinson's about protein aggregates, and they've had huge clinical failures trying to chase that down, to the point that some companies have abandoned their neuro program because of it. And so I guess I'm trying to get my head around how strong is the causative effect of these granules. Is the disruption in autophagy a symptom of the disease, or is it driving the disease? And I guess that's where I don't know, has the literature sussed that out?
Go ahead, Hemo.
Yeah, so if I may, I mean, I sort of agree with Chris, because I think it sort of goes down to the lysosomal pathway in the broadest sense. And perhaps, I mean, we as cell biologists, biochemists, we need to work out what actually the defect is, what is the problem, and there's some really good suggestions already. But as a clinician or somebody who wants to design a therapy, isn't there ways of actually then rather curing the defect of the pathway? So sort of trying to rather address, let's say, autophagy, try that. Is it autophagy? Can we stimulate autophagy? Because apparently there is a problem. Or following up on what Virginia said, can we manipulate somehow the lipid metabolism, right, to get the ceramides down? The ceramide down. And so my feeling is that therapy could go that direction rather than perhaps going after P97. I wouldn't stop that, but it definitely requires much more biochemical analysis, as people have pointed out. We don't really know what is going wrong, and if something is different in the mutants, is that actually the cause of a disease? There's conflicting results right now. It could be actually that the activity of the unfolding activity is not so critical, but it's rather the targeting. Somebody said that also, perhaps Michele. So the targeting is critical. So we need to, if we want to cure that on the level of P97, we may actually want to cure the targeting event. So that could be additional cofactors rather than e-adapters, right? I suggest that we call adapters only those that have been, now that we can do that, have been shown to really target proteins and initiate unfolding, right? So put them into the pore, that's an adapter. Bind a substrate, put it into the pore, and all the other proteins are modifiers, modulators, and targeting factors, right? and there's been a few suggested to actually be related to the disease. So perhaps this is where the therapy needs to come in and try to repair that, right? So some sort of refolding drug or something, right? So that's been shown for other diseases, the cystic fibrosis, right? But I mean, when you say that people should try to cure the defects that we already know occur in the cell, so autophagy, lipid metabolism, and people say, yes, we have so many different. Do we really have so many different pathways that are important? It could be this unknown substrate that is degraded at a particular point, and that's why myofibers degenerate. That could be, but it's more likely that it's possibly the autophagy, right? And there's a lot of things that are apparently not relevant. I mean, tell me, please correct me, but is DNA repair, DNA replication, genomic stability doesn't seem to be an issue at all in the disease, does it? I mean, so there is actually sort of, we can actually already, and by the way, the genetic or genomic stability is apparently the target for cancer drug treatment, right? So that is actually going wrong if you treat cells with the p97 inhibitors, and that kills the cells, at least some of the cancer cells, lymphoma cells. So I'm just saying that some of the things are apparently not so relevant, and we know that already for the disease, right? And as Chris said, there could be the lysosomal pathway. I think this is a really hot candidate, and perhaps we just need to go from the other side and find something to cure the lysosomal system rather than the p97.
So if I can, something that we could do that would be really helpful for the field is if you had a table. These are all the supposed functions. These are all the things that change. And then whether the genetic knockouts or chemical tools on that pathway have an effect, and then start ticking through it that way. And that's probably going to be, I mean, it definitely would be disease dependent, but it could also be cell type dependent. And if turning on autophagy at some level, it might not just be mTOR. You might have to see where in autophagy can you fix the deficit. But if you can find a way to fix the deficit and that addresses the disease, then you've turned that chemical tool now into a drug lead. So I think that's a really great way to go, and in combining genetic tools with small molecule tools where they exist or where we can develop them. It's really important. I really liked what Sifen showed, that the compensation and the difference in time course that you can get from a small molecule versus genetic manipulation. It gives us a lot of insight too. Yeah. So what can we just cross off because the genetics or the small molecule perturbations don't fix it?
Something I slightly worry about is how much of our understanding is just driven by people in the field having a certain interest, right? So is it endolysosomal vacuolar stuff because that's what we're interested in? Maybe there is a DNA damage defect, but nobody's looked yet. It's just always in the back of my mind. Right?
Sure. Well, they don't get brain cancer.
That's true. Yeah.
Yeah. So there's
But
tissues that are affected.
I guess I think about it from the human tissue, and so think about it from. So I mean, you identified a vacuolar tauopathy for VCP mutations, like you said it looks like spongiform encephalitis. There's lots of things that are similar, and so in muscle, I know other causes of vacuolar myopathies that have clear causes because they impair lysosomal degradation or because they impair autophagy. So I mean, I don't think we're missing something.
I'm on board. I'm just
No, I hear you.
Yeah.
So, if in your opinion, VCP is crucial for the fusions of lysosome and the autophagic pathway, and And if you think that that's crucial, if we activate autophagy, we are going to fail because this enzyme is crucial for that. So what do you think about inhibiting autophagy?
Yeah, so we've done that in our mouse model. We made it worse. And the reason is because it's not just as simple as activating the front end of autophagy. It's actually fixing the defect of autophagosome lysosome fusion. And so all we did was just push more through the system. It may be that we need to do another type of not just activating autophagy, but maybe it's facilitating autophagy. And what Michele said is correct. It's looking at seeing what step in autophagy is impaired and needs to be therapeutically targeted. So, but I think yeah. So it's not as I still think it's a therapeutic target, but it may not. It may take more thought than just let's activate autophagy with a drug like rapamycin or something. I think we also need better animal models in some ways. We presented an animal model, or Eddie showed kind of our animal model, which is really not the disease. It's a single, mutation. Virginia's doing her studies on a homozygous mouse model that's been treated with, high-fat diet. That's not really a great disease model. The disease models that we have are really mild. If they get disease, it's really late. Eddie made a mouse model that didn't have any phenotype until he injected it with tau seeds. The models are not that great and they're not that consistent. So I think that's another limitation.
So Chris, is that a mouse model or is it
mouse models? Yeah, all mouse models I'm describing.
Okay, so not the fruit fly, that Alyssa has. You want a mouse model.
You can ask Jesse, but my thought is you need a mouse model.
Yeah, I think this is one of the
recapitulates patients. You need one that recapitulates patients. So
yeah, especially for any type of drug development, you need a rodent model that the field agrees is a good model for the disease. And I guess one of the caveats there is if the disease is so diversely manifested, will we get to a mouse model that is consistent, or will the mice suffer from heterogeneity and not really recapitulate all the aspects or only certain aspects of the disease? And I think in the back of my mind, one of the questions I still have is the pleiotropy in VCP driving the heterogeneity we see in the disease? And is it just a random or other genetic modifiers that are causing the different disease manifestations that are seen, or the different disease progressions? And I don't know that we can figure that out in the time span of a mouse. We might have to look at case history and longitudinal studies to know that type of information.
So Jesse, what about stem cell or iPS cell modeling? Would FDA be willing to accept that? There's some precedence from ALS literature where they used it to show neuronal hyperexcitability and use drugs model to work on that.
Yeah, I think, I'm not a regulatory specialist, but I think the FDA is interested in further drug development in neurodegeneration in general, and they're kind of eager to get things that are working. I think one of the things the FDA has, first and foremost, is they look at safety. And so you have to have some mouse model that you can show that you can safely dose. And they usually like that to have an efficacy readout also. So I think you can provide a lot of cellular data that's supportive preclinically, but I think you need a mouse model that, if the FDA calls up any of the experts at this conference and says, is this a good model for the disease? You guys would say, yeah, that's the model I would use. And I don't know that that's settled on right now.
But if there's no model, you can develop it without a model. Because you're taking the risk on efficacy, the FDA is taking the risk on safety.
Safety, yeah.
I think Jesse's the right way to say that. So if the fly model or the cell model recapitulates the key point in disease, and what the key point in disease is, such that you're willing to take the risk and you can demonstrate it's safe in all these bunch of animal systems, then you can you can take that bet. The question is, can you get a company to pay to take that bet? So, yeah, I think the fields can get held back. I mean, these age-related diseases, mice don't live that long, so you have to do something to make them get diseases in a short period of time, unless it's something that's intrinsic to the clock of a lifespan. So, you throw out the baby with the bathwater by doing that. And then, so in Alzheimer's, I think we think very much about these models not as efficacy models, but as pharmacodynamic models. So if I'm trying to treat this target, is this target present in the mouse in a way that I can measure? So maybe we need to expand our mind to think about it that way.
Right. So I mean, that would be great if the mice they don't develop really any
Some
reliable
consistent
pathology. Yeah. I mean, it's not that they're not getting weak or they're not getting, demented. It's that they're not really developing, robust. If they do, you would need, 30 mice per condition, and it just becomes untenable to, do 30 mice and age them all out to 16 months. I mean, it's just, it's not that they don't show a phenotype, it's just they don't have a robust phenotype, which is really what you need for these types of studies.
So I would like to comment about the mouse model. So I mentioned about a knock-in mouse model having an attenuated phenotype. So we procured Paul Taylor's mouse before he got them to Jack's. And the problem with that mouse model is that the gene is on the X chromosome. So we can only test males. There's a lot of waste. It's quite expensive. It is a faster model. They're developing weakness around 5 months or so, but it's still a slow model. You're talking about a long study duration. So we really need a mouse model. So I know that Paul Taylor published his R155H mouse, but it no longer exists. And I'm wondering whether Cure VCP could sponsor that mouse resurrected or creating a mouse just like that, because I personally haven't actually seen a patient with the A232 mutation. It was something we published. It was a Canadian family with a very severe phenotype, and that has become historic, that it's very rare, and we really it would be better to have an R155H mutation, a mouse with that mutation, because the vast majority of patients have that mutation. And maybe there should be a series of mice, with all the other mutations also. I know the R155C had no phenotype, which was a big disappointment, but perhaps an overexpressed.
Well, we're talking about different. You're talking about knock-ins versus overexpressed models. Mm-hmm. And so I do believe the overexpression models may not be the most appropriate. And we've moved into a genetic era where making the CRISPR knock-ins is actually just as easy as making the overexpression models.
The only thing is they don't manifest. I mean, ours is a knock-in, and the R155C never manifested. Apparently.
So Virginia, doesn't Linda Greensmith have some of those Paul Taylor mice, or?
She uses the same A232, which is on the X chromosome. It's difficult to work with, but it does have a phenotype. Yeah, that's what we're using now. But as Chris says, it's an overexpressed model. It's not really like the human. Perhaps different backgrounds of the mice would make them manifest. I don't know, Chris, what are your thoughts, transforming them onto different backgrounds to see if we can tease out that phenotype, these knock-ins?
I don't know. I mean, I think our approach is going to be to try to make worse mutations somehow. So to really understand that VCP doesn't bind perhaps to UBXD1 as Hemo's shown, and to try to make VCPs that have worse binding to UBXD1 and see if we can make them worse. I don't know. I don't know if remaking mouse model after mouse model is going to do much. think we want to advance the science. I think we also want to obviously create a model that we can use for clinical testing, but I also think we also want to advance the science in some way. I don't know. I don't have a good answer.
All of the above.
Yeah, you mentioned the overexpression versus knock-in. What do you think about, or versus CRISPR? How about for cell lines? Is overexpression going to be different from heterozygous mutations at wild type levels? Is in VCP disease, are the mutants at the same level? Is it 50% at the protein level, mutant and wild type?
I mean, we don't know for sure, but yes, I
More or
think
less.
probably, yeah.
Yeah.
It isn't degraded, yeah.
Yeah. It isn't. It is degraded, or it's not?
It isn't, yeah.
It isn't, yeah. So it's like a wild type. It expresses similarly to the wild type allele.
Yeah. Having said that, the R155C mouse model did show degradation of the mutant allele. So there was more of the wild type allele in the brain particularly, but also in the muscle. And that was thought to be the reason for the attenuated phenotype. We did not see that with our R155H variant mouse. So it is a mystery.
If I may say, I would advise caution with tissue culture cells and the overexpression of the mutant form. We've done experiments and had good results. We could differentiate actually many years ago some sort of effect on the lysosomal system versus ERAD actually. But then in other conditions where we overexpress, we even had effects on DNA damage. And I don't believe that it actually has, because patients would definitely have different symptoms if that was actually anything related to the disease. So I'm just saying, I think it is a good model, but one has to have caution there if you overexpress a mutant.
Yeah.
You get nice one-to-one expression levels, but still it could be a bit dangerous that you actually induce some defects in pathways that are actually not affected in patients.
It gets that Goldilocks effect, right? So yeah, if you have too much of everything, then you hit the other side of the Goldilocks.
Possibly.
So Michele, I don't believe we need to overexpress cells, right? I mean, just for proof of concept, etc. But we have so many lines with all the different mutations and phenotypes, through Coriell. And through different folks working with iPSCs, one can get all the iPSCs. I know it's really hard to differentiate to myoblasts, but now even that has been cracked by many labs. But I think that's not a problem. The lines, there's plenty of those available. It's the mice that are a problem. Yeah.
But even with CRISPRed lines, once you make a CRISPRed line and subclone it, then you've introduced heterogeneity, right? And you could fake CRISPR it and have the wild type allele, and when you subclone it, it doesn't grow as well, right? So I think you need numbers or somehow control for that. So there are problems with every approach, I guess, and you just have to know what's good and bad.
So I hear what you're saying about iPSCs being so available, but most of the papers you see use cell lines, and same, we've got really great inhibitors of VCP, and yet you see DBEC being used, which is really, quite a bit less potent and less soluble and less selective. And so it's hard once these tools are out there to really define as a community what are the best ones. And maybe we don't want to, because then we fool ourselves because we're all looking under the same lamppost. But I think just the same things that you have in cell biology, I think about in chemistry.
One thing that I put comments in the chat was regarding the clinical trial readiness. I really believe that if we did more natural history studies, maybe had a consortium like part of the Rare Disease Clinical Research Network, that will lead to trials and more research and collaborations, etc. And I propose that our group should submit a Rare Disease Clinical Research Network proposal. I don't know how long they're going to continue that funding, but we have Tahseen and Chris and
So there was,
Jordi.
Virginia, there was a recent round of funding. Chris was very much part of it and I think we had put in an application for limb-girdle muscular dystrophy as part of their RDCRN. It did not get funded. Some of the existing sites did get refunded. I don't think it'll come up for renewal anytime soon. I think I believe it's 7 years, right, or 5 years. At least they're locked
usually
in for
4 or 5 years. Yeah.
I think they're locked in for 5 years, so that opportunity actually may not come up for another 4 or 5 years. But I think this is a great opportunity, and I really welcome the idea of a longitudinal natural history study, but inform it with biomarkers or whatever imaging studies, etc. And I think, and this really needs to be done as an international consortium with the help of the relevant groups all over the world. And so my request would be that let's start thinking about it. I've been talking to Chris about it for a while. And then I think I've sort of broached the idea with Jordi as well. But I think there's really high time that we put together a consortium, maybe as a multi-PI grant mechanism, and put a strong case to the NIH for funding it.
I totally agree. Yeah.
Yes, one of the things is that we now know is, thanks to this international collaboration, is where are the patients. So in Europe, for example, or in Asia, what are the sites or what are the centers that have a large number of patients, which is always helpful for this kind of international natural history studies. So where can we find partners that are following a lot of patients with VCP? So I think it's also interesting. So, Virginia, you were mentioning clinical trial readiness. I after listening the talks today by physios, for example, Mendez and also Lindsay, I would like to know, I don't know if they are here, but I would like to know their opinion or your opinion about, okay, let's do a clinical trial. Then we need to select an outcome measure. What outcome measure do we select for a clinical trial in VCP? So that's a very complicated. Just reviewing muscle weakness. You say, okay, let's take a look to muscle weakness. Okay, you see that the patients can start with whatever pattern they want. So proximal and distal upper limbs and lower limbs. So that's, in my opinion, one of the things that we need to know better is try to find out what is the best combination of muscle function tests that can clearly identify changes in these patients. Now, great, Lindsay and Melody, you are here. What do you think about that?
That's a great question. Never heard that one before. Just kidding.
That's the typical question.
And she just happened to be online and answer that question.
Yeah, I think that's the point of our current longitudinal study. I think we're at phase 1, so we'll have much more information in May, June, July of next year when we see how sensitive to change these outcomes are. If you made me choose today, I would, but since you can't make me.
Oh, I want to know. What if it was today? I couldn't tell from your talk. What would be the choice?
If you made me choose today, I would choose a composite sort of outcome. So not looking at them all together, but I wouldn't just select one, just because there is such heterogeneity in the population. I think there is benefit to, if we didn't have a mutation-specific treatment, trying to include as many patients as possible. So I would, to keep your ceiling as high as possible, I would include the 100-meter because patients can run, and none of our patients yet have run at a speed that would be typical. I would include the NSAD because it has a lot of meaningful functional assessments and has been functioning really nicely in this population. I would also include upper extremity assessments that are useful to kind of lower the floor for patients that may lose the ability to walk. So patients that are on the call, close your ears, but I would pick probably the pull and active, even though they hate it, because it's been nicely fatiguing. Sounds bad. I don't like to fatigue patients, but it has been more challenging for some of the stronger patients than the pull.
So you didn't name a composite, you just renamed your current study.
No, I did not name all of them.
All right.
I left out some key ones.
But again, I mean, and Lindsay, but you're thinking of a composite primary endpoint rather than individual endpoints.
No, I think it gets a little bit complicated when you have a composite primary endpoint. I do think there's ways to identify some subgroups and maybe have a primary outcome for certain subgroups. That is just one of those outcomes, whether it's the POL or 100-meter or NSAID. And I see Amy's comment. It is not the dumbest game ever. It is so good.
I love that you're commenting. I think I should exit, and one of them should come on and give some thoughts. I would love to hear from Amy or Camille or somebody.
That's a great idea.
Nathan, can you also kick me out of here so that other people can?
I think if you on the right,
I think you just have to leave.
You hit leave. So
I'll
still be here. I'm just. Yeah.
In the absence of our international collaborative funded trial, it does offer the opportunity that those centres that do have a cohort of VCP patients utilise their MDTs, because I'm sorry, but MMT doesn't cut it as a follow-up outcome measure for these patients. It's not going to drive their clinical care. It's not going to help kids decide if they need a wheelchair. So if we can harmonise what we're doing to centres other than Nationwide and Newcastle, we've got a really beautiful opportunity to make every clinical appointment count for the patient and for the greater community as well.
Absolutely.
And if the people want their PT to be trained in some of these outcomes, I'm sure Lindsay and I would be very, very, very happy to do so.
But, I'm really quite impressed by MRI as a way of monitoring changes. We did that for an exercise study, and within 6 months of exercise, we were able to see differences in a cohort of patients with Pompe disease. And I would propose that should be, I know it's expensive, but it's a good tool. I don't know what people feel about muscle biopsies, whether that is a useful thing to study in patients longitudinal.
So I would love to hear Jordi's comment, because one of the challenges with MRI is standardizing the hardware and the software and to get sort of harmonization of the images. And I know Jordi and I are on a trial together right now, and the amount of rejections of the images is very heartbreaking most of the time. I'm still recovering from the PTSD from Pierre Carlier's rejection of the Dixon imaging in our site. So how has it changed, Jordi, since then? I mean, are we able to get better harmonization of the images?
Yes, I think that it depends a bit on what is the sequence that you want to use. So, for example, for Dixon, I think that we are now closer. Dixon is useful for those that are not a specialist in an MRI. Dixon is useful to identify and quantify exactly the amount of fat that is present in the muscle. And now I think that we are closer to have a better standardization of Dixon across different sites. So that's not so far from here. I think that, well, in the United States, you have a good example with Imagine DMD, this consortium of sites assessing patients with Duchenne, that they are doing MRIs to many of them, and then it's a good example. The point is, if you want to use other sequence, for example, if you want to analyze inflammation or active necrosis, that we use in general T2, and T2 it's more complicated. Even having you probably will need a patient go into all sites and just to have a clear and doing an MRI at all sites just to standardize the measurement. So it's far complicated. So, and I think that what we could expect is an early change in inflammation and necrosis after receiving a treatment, so with T2 normalization, and in the long term, reduction in the increase in fat replacement of the muscle. But we still don't know what is the ratio of increase in fat replacement in patients with VCP. So, and we need to compare. We can compare treated versus not treated, but it's not so easy because the velocity of fat replacement depends on many different factors. And one of the main factors is the amount of fat that you have at baseline. So you cannot just compare treated versus non-treated. You have to compare treated versus non-treated and patients that have more or less the same amount of fat at baseline. So I think we are far away from using MRI in VCP because we still need to do a lot of studies in cohorts. So I am more than happy, for example, to see natural history studies, including MRI in VCP.
And Virginia, in muscle biopsy, are you looking for morphological changes, or is it going to be mainly biochemical changes? Because, I mean, especially with the supercore needle biopsy that we are using for some of the limb-girdle muscular dystrophy studies, it's a relatively atraumatic biopsy, and can be done much more easily. So that's something that potentially is doable, but it may not be able to be used for morphologic analysis.
Just biochemical studies.
I mean, again, it's just we haven't tried that, and I don't know if Chris has tried it or not, but at least, I mean, you get about 30 milligrams of tissue per pass, so it's a fair amount of tissue for biochemical analysis and molecular analysis, but we haven't tried fixing it for morphological analysis yet.
Yeah. I wonder if EM might be a better test with tiny amounts. I don't know.
Well, I mean, again, but it's the same issue of fixing it and orienting it for. I guess, I mean, that's something that we can look into.
All right, Amy, muscle biopsy or active?
Me? I don't know, but I do have a question. Is there a place for storage from live tissue donors? If people volunteered to donate live tissues, is that something that would be beneficial? And is the Brain and Tissue Bank responsible for that, or do they only store cadaver tissues?
So I can answer the first part of the question. Of course, it would be beneficial to have access to tissues from patients. That's for sure. So as I am working in Europe, I don't know how this can be done in the United States. I need help from others, but for sure it would be beneficial.
Yeah, I always ask people if they're having surgery, let me know, because I would then coordinate with taking, what would be normally discarded tissue to collect, because it's so valuable having living tissue as opposed to, something that's two days old even. But we're talking about, muscle or a piece of bone, obviously. But some people have even had brain biopsies, which we actually were able to retrieve and study years later. So yeah, living is always better.
That's very good to know.
Shoot, because I have a lot of tissue on my belly.
Well, it's actually a good source for iPSCs, for stem cells.
I have a question. So would you be thinking that like MRI over an EMG would be more helpful down the line? Because, for me, that EMG, that muscle test, it really left my left thigh way weaker. I mean, I had a lot of problems after that and continue to have a weaker left thigh. But
so I think that these are two different tools that measure different things. So MRI is useful to look at the structure of the muscle and can be helpful in driving diagnosis, can be helpful in following up the disease. EMG is useful, especially useful to explore the function of the peripheral nerves. If we want to analyze or quantify the amount of damage in the muscle just with EMG, that's far more complicated. So EMG can be useful for the diagnosis process of someone that is weak, and then can help us to distinguish between a neuropathy, a myopathy, etc. But I don't really see EMG as a tool to follow up patients, for example, especially with a myopathy, where there is nothing that really objective that you can measure, that you can put a number, for example, to that. So I think that these are different tools that have different utilities.
I would like to second what Amy said too, is I think, or emphasize it, is that there's such a big population of patients that are willing, I believe, to give, samples, like if it's blood or skin samples and things like that. So, just reach out to our community, because I know I'd be in line to help out.
Yes, I think it's also good to know if you have in your community people from UK. In UK, there are also other good opportunities to donate tissue. There are the MRC-funded biobanks in London and in Newcastle, for example, and patients can just address to us and say that they can donate and they want to donate tissue. I think it's something that we are storing serum, muscle, skin, fibroblasts, etc., in the biobank. So I'm sure that there are opportunities in almost each country.
Yes, it's been on our action list. I know Lindsay, as part of the natural history study, collected samples that I think Tahseen and Chris are going to store, right? But we can do more.
Yeah, I would be, Lindsay said, muscle biopsy. I would go through it. I would go through it for this. I would be willing to do whatever I have to do. I come from a long line of people in my family that had this, and I have 4 kids. So whatever I can do for them, I will do.
Nathan, this probably should be planned because I think collecting is one thing, and then making sure that it is collected in a IRB compliant and potentially, and we have all the agreements lined out in terms of sharing the data, especially if it's going across continents as well. But I couldn't stress more the importance of having a biorepository like this, where you could be collecting muscle, skin, or whatever it needs to be done.
I have a comment about the muscle collection. So when my dad was early on in this disease, before we even knew that this was a disease, they were thinking he might have another disease, and they collected muscle biopsy, and it just happened to be a collection of muscle that wasn't affected. And so they said, oh, you're fine, you're normal. And then, years later, they did another muscle biopsy. They happened to get stuff that had some of those, aggregates and proteins and so forth, the yuck in it. And so it happened to be misrouted and sent down to Mayo Clinic, and they identified it as inclusion body myositis at the time. So my concern would be is I don't mind donating muscle, but might I be too early on in the stages that you might not get any of what you're looking for? How do you find that? And, how would you muscle biopsy the spot that you would need? Is that where you do maybe an MRI and say, okay, some of the yuck is located right here, let's biopsy that?
Yeah, that could be helpful, what you are suggesting. MRI can also help driving the muscle biopsy, so guiding where we have to take a muscle biopsy. And of course, it's a good suggestion, and it's something that we also use in patients that only have increased levels of CKs, for example, and we want to do muscle biopsy for diagnosis. You don't really know what muscle could be the best one. You can use MRI, and here in our country, Europe, I used to do MRIs just for guiding the muscle biopsy. It's not a problem.
So we have 5 minutes left. Tahseen, you kind of hit the nail on what I wanted to do is try to identify. This was a phenomenal discussion. I mean, this was great. This is exactly what I wanted to happen. But what are the specific things, kind of leaving, just like the ENMC meeting in 2015, there were specific actions identified that needed to take place. So one of them, Tahseen, is we need to find a tissue bank or something that patients can donate, because that will help the scientific community. I also heard mouse model, though it was a little, how exactly, does it use CRISPR? Can it replicate? But and for our audience that's, there, please, in the chat, or we've got 2 spots on camera. What are those specific things? What do we need to go drive? And again, Cure VCP Disease, we'll go help what we can. Virginia, I know you mentioned the CDRN. I think that's pretty ambitious, and probably not likely at this time. But identifying biomarkers. I mean, some of that just sounds like time. I mean, we just got to have more time. Certainly Lindsay with the study that we're doing, there's going to be time. Jordi with your study, it's just going to be time. But what else? So I've got those 2, mouse model, tissue donation, how to collect those tissues. And then, Tahseen, you identified this kind of consortium, global, putting that together, which I know that's been on your radar, your list, working together to try to get that funded. So are those the 3?
So again, Nathan, what and I don't know how many patient families are on the conference call, and unfortunately I was in Chicago all this time, so I missed most of it. But I think one thing I wanted to emphasize was that there's no lack of interest and there's no lack of enthusiasm about a drug study. But there are many challenges to launching a successful drug unless you understand the disease, unless we have better outcome measures, unless we have good biomarkers. It's going to be very difficult to run a successful study, and I'd rather not sort of waste energy or efforts or even resources on a study that is probably going to be doomed to failure. I think the work that Lindsay is doing, the work that Jordi has been doing in terms of MRI images are very important. Virginia has a lot of history and a lot of previous experiences with identifying potential biomarkers. I know Chris has been working on it as well. I think it's time that we make a concerted effort to develop a patient bank, maybe through 1 or 2 centralized sources, have some kind of a sharing agreement so all the researchers can share it, but then at the same time, do it also as part of a natural history study where we can do serial MRIs or whatever biomarkers we need to do. It really has to be a multinational effort. You're not going to have enough patients in any one country, any one continent to be able to do this. So maybe working with the European colleagues and the South American colleagues or even Japanese colleagues coming up with an agreement on that.
So you got that action?
I have that on my list. We just need to hash out some of the agreements there. And I know Lindsay is smiling because Lindsay wants to help me with that.
You know it.
and I'm only looking for an excuse to visit Newcastle. So Jordi will have to host me for that. So
you can come whenever you want. You will enjoy the weather and the food.
And the cricket matches. Remember,
the cricket's on in Yorkshire this week, Tahseen. You can come and watch Yorkshire play.
Okay, this session's deteriorating now. We're talking about cricket.
Hey, there's nothing wrong with cricket.
Okay, was there any other comments or questions from the group? We've got 1 minute, and then Sujata's gonna close us. But again, I thank you for everybody jumping on. I hope I found it valuable. I know others did. This was the purpose of what we wanted to do. So and I know some others have spoken in the chat. On behalf of all the patients and care partners, we appreciate every single one of you that is on this, that participated in this conference. We really do. So, all right. Anything else? Okay, Sujata.
Hi. Okay. All right. Well, whoopsie. I'm sorry. I'm technologically impaired just in these 30 seconds here. So welcome to the just the closing remarks here. Wow, these last few days have been a whirlwind of information and thought-provoking discussions about VCP disease, and all of that culminating to session 9, which was really incredible. I mean, that's exactly what we wanted to happen in this space. We're thrilled with the level of engagement and discussion that we were able to facilitate here. Dr. Arkin stated earlier that the more she learns, the less she knows, and that seems so true. It seems like maybe we've come away with more questions than we had before the conference began. And while there's so much left unknown, I think we can all agree that this monumental scientific conference has been incredibly valuable in defining where we currently are and some possible directions to pursue to get us closer to our ultimate goals. So just a little bit more housekeeping. If you still haven't had a chance to visit the posters and the abstracts in the expo, the great news is that the expo will be available for your viewing pleasure through 8 p.m. tomorrow, Eastern Time, and tomorrow, which is Saturday, September 11th. The researchers who submitted them put a lot of time and energy into their research and in getting their presentations together. There's some really great information there. So for those of you who submitted posters and abstracts, thank you so much for your contributions. And as a reminder, the prize winners will be notified by email and social media on Monday, so be sure to watch for that. Also, the recordings for this entire conference will be available on this Hopin platform until tomorrow, which is Saturday, until 8 p.m. Eastern Time. So if you missed a session or you want to revisit a session, you can do so by logging into your Hopin account and then clicking on the replay button on the left side of the screen. So, the conversations and collaborations that we've begun here, they don't end here. We had a lot of information shared, and truly, I wish we had more time for informal discussions. Hopefully next year, we'll be able to host this conference in person, and we'll get to meet one another and have lots of late night discussions. But in the meantime, let's keep the conversations going and use this conference as a springboard as we move forward with VCP research. And then finally, we all share a common goal, and that is to ultimately find a treatment or a cure for this disease so that generations to come won't have to bear this disease. So to our researchers and clinicians, I'm a big baby here, I'm tearing up here, but to our researchers and clinicians, you are the hope of the VCP community. And we thank you from the bottoms of our hearts for your dedication to your research, your participation in the scientific conference, and most of all, for using your brilliant minds and compassionate hearts to continue to move towards further understanding of this disease, providing hope to countless families. So once again, thank you to all our panel speakers, our researchers, our industry partners, and our patients for joining us and participating in this conference. Thank you also to our financial sponsors, which are the MDA, the Muscular Dystrophy Association, All Stripes, Every Life Foundation for Rare Diseases, and Cure VCP Disease. So at this point, the first ever very exciting North American VCP Scientific Conference of 2021 is officially ended.